Any questions before we start? Okay. That makes it easy. So keep working on the Socrative assignment if you can, if you've got internet access. If you don't, I'll leave it open for a few hours. So if you want to practice, show that you've been here, that's totally fine too. So I wanted to start with a summary from last class. Do I say "so" a lot? Did this turn into a drinking game? Is it okay? I had a professor, like a chemistry professor, that had some sort of nervous habit or something, and every time he said something, we'd make a little tally mark. I'm wondering what mine are. You probably won't tell me, but I'd love to know. Maybe at the end of term evaluations, you'll tell me what I do that's annoying. So, summary from last class. See, now I got the conversation going. Now everyone's talking about me. My ears are burning. Last class, we learned that linked genes, thank you. Linked genes don't produce the 9:3 to 3:1 ratio that Mendel would have predicted. That's our expectation. Always independent assortment. If it's a dihybrid cross, like A over A, B over B, a dihybrid, if you self that dihybrid, that produces that 9:3 to 3:1 expectation. Bateson and Punnett did this and didn't see that 9:3 to 3:1 ratio. So they were the first ones to start thinking about when genes are physically associated on the same chromosome, that might change our expectations. How does this change our expectations? We have to talk about parental types and recombinant types. So if we don't have independent assortment, who do we see more of and who do we see less of? If there's linkage, if there's dependent assortment, if alleles are stuck together, then we see more parentals. We see fewer recombinants than we would expect because those alleles are stuck together. They don't get separated as often as we would have expected. And how do we know how likely it is that two alleles that are physically on the same chromosome get separated by crossing over, by recombination?
Depends on the distance.
Yeah, how physically far apart those genes are dictate how likely it is that crossing over happens between them.
Dr. Hutt?
Yeah.
When there's like people that look like a lot like their parents, is because the parent had like genes that were like really close together, that way they didn't have a lot of recombinant.
Oh, that's an interesting question. So when you see offspring that look a lot like their parents, is that because there are a bunch of genes that are linked that control those traits? Possibly. Not necessarily, but definitely possibly. And we'll look at an example of that next class. Although it's probably not what you're thinking of. It has something to do with upset stomachs and diarrhea or something like that. But we'll get there. So we can use how frequently we see recombination happening between genes as a measure of distance between genes. Okay. And there at the bottom I've summarized some things we agreed on, or I told you we were agreeing on last class, that there was going to be one tetrad undergoing recombination per meiosis and so forth. So you can read those. I said there was going to be a chi-square test, so here's the chi-square test. And this chi-square analysis will be on the next test. It's really critical, not only for this class, but for other classes you will probably take. So if you're not already a chi-squared expert, here's a chance to practice. We talked last class about, here's Bateson and Punnett's data. They did just what I drew on the previous slide. They had a dihybrid, they selfed it, and they didn't observe the 9:3 to 3:1 ratio that was expected down here. Instead, they observed something that was like an 11 to 1 to 1 to 3 ratio, which we calculated last class. So how do we know whether or not their observed and their expected data are different? Are they different? They're not the same. So we're going to use a chi-square test to distinguish, do the observed and the expected values match or not? So somebody remind me, chi-square test. what do? What's the formula for a chi-square test?
Observed minus expected squared divided by expected.
Right. So for example, 296 on the top line of that table, 296 observed minus 240 expected, square that, divide that by 240. Then what do we have to remember to do? We've already done the formula for how many lines in that table? Well, we've used one of the lines that are in this table, the top one, purple flowers, long pollen. Now what do we have to do? To do, yeah, purple flowers, round pollen, we have to do observed minus expected squared over expected. So we add the same formula for the next phenotype. Then we have to add observed minus expected squared over expected for the third. And we have to add that to whatever the calculation is for that fourth set of data. So we've got 4 different phenotypes. We've got purple long, we've got purple round, we've got red long, and we've got red round. There are 4 different categories of F2s that we're looking at. So once you do all of these calculations, you get the chi-squared test statistic value. In this case, I'll let you work it out if you like to practice. It's 115. So once you do observed minus expected squared over expected 4 times, add them together for these 4 different data sets, it's 115. That's the chi-square value. What's the other thing we need in a chi-square test? We need degrees of freedom. So how many degrees of freedom are there in this case? Number of categories. In this case, we've got 4 different terms in that chi-square. We've got 4 different phenotypes. So what's the degrees of freedom? It's number of categories minus 1. So in this case, it's 4 different phenotypes. There are 4 lines in this data table. Minus 1 is 3. So we have 3 degrees of freedom. We have a chi-square value that's 115. Here's the chi-square table. What's the p-value that goes along with that combination of values? 115 is the chi-square value, and the degrees of freedom is 3. Well, let's see. The first part's easy. We focus our attention on the row in the table where the degrees of freedom is equal to 3. What are the numbers that are boxed in red here in this row of the table? What are those? Are those p-values? Or are those chi-square values? Those are the chi-square values. So where does 115 land in this range of chi-square values? 115. Everybody point to which end of the table 115 is for that row. Everybody should have their hands up and pointing over to that side. It's way, way, way off the table, right? We've gotten to 12, chi-square value of 12 here. We're looking for 115, way the heck over there. So what's the p-value? How do we tell what the probability or p-value is of having a chi-square value that large? What if we had calculated a p-value of 12.838, or a chi-square value of 12? What would be the p-value? That's where you look up here in the header of the table and look at that subscript. So if we have a chi-square value of 115, is the p-value really big or really small? Really small. In this case, the p-value is something like, let's see, 8.64 times 10 to the minus 25. So that's a 1 in 864 octillion chance that our observed and our expected values match. So when p is less than 0.05, we conclude that the observed values are not equal to the expected values. in this case, the p-value is infinitesimally small. So there's basically no chance that the observed and the expected values match, which is maybe what you would have concluded anyway, just by looking at the observed and the expected numbers of individuals in this F2. Questions about chi-square testing? Just one more example. So in this case, this proves that Bateson and Punnett were onto something. They had discovered linkage. Yeah.
Why did we need to calculate the degrees of freedom again? Why did you use that 3 for?
Why was it 3?
No, why did we need to do that?
Better ask a statistician. So I could answer it, but it's a complicated answer and I don't want to diverge too much. So let's talk afterwards, if you're willing to. So you have to calculate the numbers of degrees of freedom just because the p-value depends on the number of degrees of freedom. So it's a statistics debate we have to have about why is degrees of freedom important in general in statistics. It's much deeper than we need to go into in class.
For the problem itself, we could have just gotten 115 without needing that.
That's right. You calculate the 115 chi-square value without knowing the degrees of freedom. Degrees of freedom is just the number of phenotypes or the number of categories you have minus 1. Okay. So today's goals for this section of this chapter are to figure out what is a test cross. It looks from the Socrative results like that's definitely something to cover. We've already used the chi-square test. We're going to calculate genetic distances, and that all comes together. Why we care about calculating the genetic distances again, we want to figure out where genes are located relative to each other. It's all motivated by the question that we started asking last class. How do we know if we can combine different phenotypes from different individuals by crossing them together? So we want to know where genes are located. Are they on the same chromosome or are they on different chromosomes, and how close or how far apart from each other are they? And that's what genetic mapping is useful for. So here's a physical representation of a chromosome. 2 double helices, must be mitosis maybe. Okay. That's a physical representation of a chromosome. What's a genetic representation of a chromosome we could draw? It's physical because this chromosome is something we could look at under a microscope and see something that looks like this, a metaphase chromosome, pair of metaphase chromosomes. When we're talking about genetics and we're talking about recombination, maybe we want to think about what's happening to a tetrad at meiosis, because this is when recombination is happening. When we're talking about genetic representations of chromosomes, we're talking about tetrads at meiosis, when crossing over happens. So let's say those 2 sister chromatids, 2 and 3, are involved in a crossing over event. The 2 sister chromatids break and switch parts. That's a genetic representation. Why is that a genetic representation? We're still drawing things that look like chromosomes. But crossing over is the definition of representing chromosomes in a genetic way. To make this a little bit more explicit, because I know this is all very vague, what's the unit of measurement we use when we're talking about physical chromosomes? If you want to say how far apart are 2 genes, and you're going to use a unit, they're 6 something apart, what unit are you going to use for physical representation on the left? How can we measure physical distance between genes? What's 1 unit of DNA?
Map unit.
So map units, we're talking about genetic side. What other terminology do we use for genetic distances besides map units? Which one? Base pairs. So that sounds like a physical distance, right? A building block of DNA is a base pair or a nucleotide. So we can measure physical distances between genes in base pairs. You could just as easily say centimeters, but we could have some discussions about whether or not centimeters is a useful way to measure genetic distance. It's a physical measurement, right, centimeters? But base pairs, so you could say two genes are 800,000 base pairs apart, or 8,000 nucleotides separating them. What other terminology do we use on the right side for genetic distances, aside from map units? Reading from this chapter. Centimorgans. And the reason that I put this up here is because you must not confuse centimeters with centimorgans. Morgan was a person, so he gets a capital letter. So C with a capital M is centimorgans. C with a little m is centimeters, and totally different. Okay. There's one other that we're going to talk about, recombination fraction, RF, for genetic representations. So we represent distances between genes on physical maps in physical distances, numbers of nucleotides, but on genetic maps, map units, centimorgans, recombination fraction. All of those terms for genetic distance measure crossovers. It's how many crossover events happen between two genes. So it's just another way of measuring distance. We've got physical distance, we've got genetic distance. It's kind of like how you could measure time. So you normally measure time in seconds, minutes. In genetics class, you measure time by how many times I've said so. Right? Just a totally different way of measuring something. We've got physical distance, we've got the number of nucleotides between two genes, or we've got genetic distance. How many times do crossovers happen between those genes? It's just another way of measuring distance. I'm going to skip this slide. We'll come back to it at some point. We've talked briefly about parental and recombinant types in the past, a couple of classes ago, but this is really critical. If you're going to do genetic mapping, this is where to really be paying attention. Okay. What's the top generation up there? Big A over big A, big B over big B, cross the other one. What generation is that? Yeah, this is the P0 generation. Okay, so that's the F1 generation. Here's the F2 generation. And all of these things that are drawn up here are genotypes. There's slashes. So we're talking about diploids, we're talking about genotypes. If we're going to measure crossing over events, which is how we're going to measure genetic distance, we have to look at gametes, because that's the output of meiosis. When does crossing over happen? During meiosis. So if we want to know whether or not crossing over events are happening, we have to look at the output of meiosis, which is what? Yeah, the gametes. So what are the haplotypes of the P0 gametes? If a big A over big B, big B over big B, P0 individual produces gametes, what are their haplotypes? Remember what a haplotype is? What's a haplotype?
Genotypes of the offspring.
Genotypes of, what's the haplo part? One copy of everything. So what cells do we find haplo somethings in? What cells do we have that are haploid? Our gametes. So what's the genotype of the gamete that's produced by that P0 individual? Big A, big B. And don't forget the semicolon. What about the other P0 parent? Little a, little b. Those are the parental haplotypes. Now, how are we going to find out if recombination occurs between gene A and gene B? What gametes do we have to look at to know if big A and big B have been separated by crossing over? We want to look in the F1's gametes, because that's the output of recombination in the F1. So the F1 organism that's sitting there, their germ cells go through meiosis. That's where we know if recombination has happened. So we don't know what the haplotypes of its gametes are. That's what we need to experimentally determine. So that's what we're trying to collect evidence of. That's the prize right there. What are the haplotypes of the F1's gametes? You could take their sperm and eggs and sequence them. That's one way to do this. We have a slightly easier way. While we're at it, though, what do we call this F1 genotype? This is a what sort of hybrid? 2 loci, dihybrid. Who's this? This genotype we call a tester. Yeah, this is so the tester is always homozygous for the recessive alleles. So little a over little a, big little b over little b, whatever, tester individual. So this is a dihybrid test cross. Heterozygous, one individual is heterozygous at all of the loci we're interested in. The other parent is a tester, homozygous for all the recessive alleles. So if the parental haplotypes are up there underneath the P0s, what are the recombinant haplotypes? If big A and big B together are parental, and little a and little b are parental, what are we going to look for in the F1's gametes that would be recombinant?
Big A, little a, big B.
Has to be, so if there's a big A, if it was with a little b, that's a haplotype that's not present in the P0 generation. What's the other one that we're missing? Little a, big B. So those are the recombinant haplotypes. They're the parental haplotypes. The parental haplotypes are the gametes, the genotypes of the gametes that the P0 parents produce. Other combinations are recombinant. They're recombining the alleles that were originally present in the P0s. So that's parental and recombinant haplotypes. When we're talking with 2 genes, there are 4 different combinations. 2 are parental, 2 are recombinant. Questions at this point? So why do we care about parental and recombinant?
They would let you know if the genes are linked or not.
So if you've got a haplotype that's recombinant, what happened during meiosis that produced those gametes? Crossing over. So we detect crossing over events by looking at the haplotypes of the gametes that are produced by meiosis. For example, here we go. We've got 2 genes that are unlinked. Yeah, go ahead.
Wouldn't that just be testing independent assortment if they're unlinked? And then if they were linked, it would be crossing over?
Right. So in the case where, which is this one, when we are saying in advance we know these genes are on separate chromosomes, how much linkage do you expect to see? There's no linkage. So you expect to see, and this is a question that I left you with at the end of the last class, if there's no linkage, what fraction of the gametes are recombinant and what fraction are parental. So that's what we're going to do right now, is figure that out. So if Mendel is correct, and if you're dealing with genes that are on separate chromosomes, which is the goal, to figure out if your genes are on the same chromosome or not, what's going to happen? So we've got the parental types, big A with big B and little a with little b. And then we have what kind of cross again? It's the exact same cross as on the last slide. We've got a dihybrid genotype. Who's on the right? All together now. Okay, on 3. 1, 2, 3, tester. Oh, man, it's not that bad, is it? So, dihybrid test cross. Now, how do we know what the genotypes, the haplotypes that are produced, what are the F1 haplotypes? How can we figure this out? We've got the genotypes of the F2s. Here they are. Talk with your neighbor for 2 minutes. See if you can figure out how you're going to guess, infer, what haplotypes this individual is producing. Okay? We'll come back together in 2 minutes. It better get a lot louder in here. Go ahead. How are you going to figure out what's in that blue circle? You've got all the data you need. You just have to work it out.
When you have the semicolon, you're talking about 2 different chromosomes, but they're from one parent. That's right. So when you're looking at the genotype, you're just looking at on the left-hand side, out of the 2 flashes, you don't get A. Right? On the right-hand side of the semicolon, you can only get B.
From the parent on the right.
From the parent on the right, yeah. So that's why you have A or semicolon B.
That's right.
And then you make a combination between the left-hand side and the right-hand side.
Right.
If it's recombinant.
Right. Or if it's not. Well, depends. Okay. So who figured it out? How many big A, big B gametes did that F1 individual produce? Better yet, you've got just enough information on this to figure out how many of each type of sperm or egg that F1 dihybrid produced. What's the haplotype produced by the tester? What gametes does it produce? Has to be 100% little a, little b. Okay. So in the F2 offspring that are produced by this tester, this is why we use a tester individual. We know that that little a in all of those genotypes came from that parent, the tester parent. right? Tester parent can only produce gametes that have little a's. So any of their offspring that have little, we know at least one of their little a's came from the tester parent. What else can we cross out up here? The little b's, because we know that in every one of the F2 offspring, it had to get a little b from the tester parent. So what's left? What's left here in the F2 genotypes? The F1 haplotypes. These are the genotypes, big A with big B, little a with little b, big A with little b, and little a with big B. Those are the gametes that the F1 dihybrid produced that fertilized the little a, little b tester gametes to produce these F2s. So how many recombinants? Is this first haplotype, big A with big B, is that parental or recombinant?
Parental.
Why is it parental?
It's one of the haplotypes that were given out by the Biol.
Right. So the P0 generation, there were big A's and big B's in the gametes, so that's parental. Where's the other parental haplotype? Little a, little b. So we've got 250 plus 250, we've got 500 parental gametes produced by that F1 dihybrid. What about recombinants? Big A's with little b's and little a's with big B's. How many F2s total? Also 500 in this case. Okay. So calculation of genetic, that's all you have to do to calculate genetic distance. You just want to know how many recombinant gametes were there, how many parental type haplotypes were there. So how do we calculate genetic distance? It's just as easy as adding up all of the recombinant types and dividing that by the total number of observed gametes. So in this case, there are 2 recombinant types that totaled 500 offspring. That's that 500, the sum of the recombinants. How many total offspring did we observe? F2s? 1,000. So what's the genetic distance between gene A and gene B? .5. This is, when you do this calculation, this is the recombination fraction, because it's a fraction, right? So these two genes are 0.5 RF apart. These are unlinked genes. We said going into this question that they were on separate chromosomes. So unlinked genes will always, this is the part to really make sure you write down if you haven't already. Unlinked genes will always be 50 centimorgans apart, 50 map units, 0.5 recombination fraction. This is what happens when your genes are independently assorting. Look at the Punnett square that's up here. We've got parental gamete, recombinant gamete, recombinant gamete. What fraction of the squares in the Punnett square for this cross are recombinant? 50%. What Mendel would have predicted. So 50 centimorgans, 50 map units, 0.5 RF is what you get when Mendel's expectation is being met, when there's independent assortment. If I've got internet access, I'd like you to very quickly do this. Draw me a picture of a physical map that is, if you were looking at chromosomes, physical chromosomes, not genetic representations of chromosomes, draw me something that represents two chromosomes with two unlinked genes. And send that back to me. Got a few minutes for this. So a difference between linkage and unlinkage. This is going to play an important role tomorrow. Wednesday. This is really critical. So as simple as you want to make it. Doesn't need to be fancy. If you've got extra time, feel free to work on the Socrative quiz if you didn't get to do that at the start of class. Here's one. What genotype? What kind of a genotype is this? Big A over little a, big B over little b? Dihybrid. Unlinked genes. Right. Two alleles of two genes on separate physical pieces of DNA independently assorting during meiosis. This is not the only way, and this is something we'll come back to next class, this is not the only way to get genes that are 50 centimorgans apart. So I just tried to make sure that everybody wrote down that unlinked genes will always be 50 centimorgans apart. That does not mean that genes that are linked physically on the same chromosome cannot be 50 centimorgans apart. So this is one representation of unlinked genes. There's another way to draw this, and we'll draw that next time. So I started throwing out centimorgan recombination fraction. What's the other term? Map units. So the first thing we calculate is a recombination fraction. That's the number of recombinants, the number of recombinant F2s over the total number of F2s. How do we convert this to map units into centimorgans? I really do honestly and very sincerely apologize on behalf of my profession. We're giving you three very closely related units that we use interchangeably. So it's regrettable. I wish we had standardized on one, but we didn't. So 0.5 recombination fraction is, how do we convert that to centimorgans, and how do we convert that to map units? Recombination fraction is also known as a Morgan, capital M. So how do you convert? Let's imagine this is a little m. How do you convert that to centimeters? If I tell you something is half a meter long, how many centimeters? Pardon? If I have something that's half a meter, 0.5 meters, how many centimeters is that? So how did you do that calculation? Multiply by 100. So number of meters multiplied by 100 is number of centimeters. Same thing's true for recombination fraction to centimorgans. You multiply recombination fraction, which also goes by the name of morgans, by 100, and you get centimorgans. So if 50% of your gametes are recombinant, which is what Mendel would have predicted, that's independent assortment, then you calculate a recombination fraction of 0.5, which is the same thing as 50 centimorgans. So it's just multiplied by 100. Recombination fraction multiplied by 100, that gives you centimorgans. What about math units? This is the most ridiculous thing I've ever heard. They're the same as centimorgans. What the? I don't know. I'm recording this. I probably shouldn't say that. So you multiply your combination fraction by 100, you also make math units. So centimorgans equals math units. At some point on some tests, at some point I'll probably give you one calculation in centimorgans and one in math units and make sure you've been paying attention, but they're the same thing. They're all measurements of how frequently do recombination events happen between two genes during meiosis. Okay. So we'll walk through one more mapping example. Some real data, not made up data that happened to nicely work out to some magical number. What are the parental haplotypes here? What are the parental haplotypes? There's going to be two. So big A, big B. Now, how has our nomenclature changed here? I'm using different punctuation now than I ever have before. Got this big fat dot in between the A and the B. That's what we do when we don't know. This is what we do in the lab. We don't know in advance whether or not genes are on the same chromosome or on different chromosomes. We can't use a semicolon. We can't use absence of punctuation. We have no punctuation, they're on the same chromosome. If there's a semicolon, we know they're on different chromosomes. So we have to have a third option for we don't know yet. That's this dot. So those are the parental types, which means the recombinant types are going to be big A with little b and little a with big B. Here's where you put that hard work to use. Here you see the genotypes. We collect all of the F2 individuals, every single one of them, and we genotype them. We've got 4 different genotypes, and the number of F2s containing each of those 4 different genotypes. Which of those 4 are the recombinants? Which of those 4 were produced by recombinant F1 gametes? What's the first step? What do we know about the tester individual? Tester individual produced little a, little b, and gave one of those to every single F2. So we know that those little a's and little b's that are in those genotypes all came from that parent. So which are the recombinant genotypes? The bottom 2, the big A with the little b, and the little a with the big B. How many total F2 individuals had recombinant genotypes? Oh, see, I did fudge the data. I made it easy. So and this still is looking at a total of 1,000 F2s. So if you've got 100 recombinants and you looked at 1,000 total organisms, what's the recombination fraction? 100 over 1,000. That's a crazy looking comma that looks like a semicolon. Recombination fraction, 0.1. 10 centimorgans, 10 map units. So the closer together genes get on the chromosome, the less likely recombination happens between them, which means you see fewer and fewer recombinants, which means the map distance goes down. So genetic distance gets smaller as genes get closer together. What would Mendel have predicted in this case? If you looked at 1,000, if you did this cross, and you looked at 1,000 F1s, Punnett square, the test or individual only produces little a, little b's. We have 4 possible dihybrid gametes. So what fraction of the F2s should have been parental if the genes are on separate chromosomes? Punnett square. So we should see 25% that have big A, big B, and 25% that have little a, little b. So that's the parental type, the parental type. So there should be 50% parental types and 50% recombinant types if independent assortment happens. If we look at 1,000 individuals, if that's our expectation, how many of each of those 4 genotypes should we have seen? What are our expected numbers? Not 442, 458, 46, 54. We should have seen 250 of each. Then you could do a chi-square test if you wanted. We're not going to do that. But this is very, very different. This number of observed F2 genotypes is very, very different than what Mendel would have predicted if the genes were unlinked. So are these genes linked or not? Map distance is 10 centimorgans. Genes are linked. We've got very different observed and expected values. Okay. So that essentially answers these next 2 slides. We won't go through these in elaborate detail, but you can work these if you'd like as practice. So I want to make sure that we get to the summary. So when you want to ask, are 2 genes on the same chromosomes or different chromosomes? There's a range of map distances we can calculate. What's the biggest map distance? If genes are unlinked, how far genetically apart are 2 genes? How many centimorgans? It's the one we calculated today. It's 50. So when you see, that was when I said, write this down. When genes are unlinked, when they're on separate chromosomes, you will calculate 50 centimorgans genetic distance. If the genes are absolutely right next to each other on the chromosome, what's the probability that recombination happens between them? That the chromosome breaks right there and recombines with another sister chromatid? Not high. Sure, we'll go with that. It's very, very low probability. So if 2 genes were physically absolutely right next to each other, maybe even on top of each other, which we can talk about if you're confused or concerned about the concept of genes being on top of each other, you would calculate 0 centimorgans, 0 recombinants, 0 crossovers between 2 genes. So this is the range we work in when we want to ask those questions like how far apart genetically are 2 genes. It's going to be somewhere between 0 and 50 centimorgans. So we've got a couple of practice. This is, please, please, please do these. This is exactly like what we just did. So you should be able to apply what we've learned today to do this question, genetic distance between gene A and gene D, a new gene that we haven't talked about yet, gene D. The one on that is going to help us have a really important discussion next class about determining the order of genes on a chromosome. This is 3-point mapping, deducing the order of 3 different genes, gene A, gene B, and gene D in this case. And that's where we're heading next. So after you're done calculating the genetic distance between, what was it on the previous slide, A and D, try your hand at this. Try to draw, like we did today in class, draw a physical map, draw chromosomes, draw where you think genes A, B, and D are. just based on what you know about the genetic distances between the genes that we've calculated. We did A and B in class today. We did A and C in class today. You do A and D on your own. Try to draw a representation of what the nucleus looks like. And I'll see you next time. Office hours right now. I have a big problem. I have pumpkin spice Hershey Kisses and I don't want to eat them all, so please come eat some Hershey's Kisses.